Okay, hello, we are from group 2. Today we'll be discussing about practical 3. So in this practical 3, we'll be learning surface sterilization of leaf explants, and we'll be discussing the result and also discussion. Okay, so for the data recording, who would like to try the results?
Since I have the result, then I'll go try.
Okay, sure.
For both sterilizing agent, it will be the same, the 10% chloride solution, but will be the different for exposure time, which is a 5 minute and 15 minute respectively. And then for the 10% chloride solution with 5 minutes of exposure time, we have prepared 5 explant for every experiment, and the number of contaminated explant for the 5 minute of 10% chloride solution is 3 per 5. So the percentage will be 60%. So the remaining number of viable explant that is remain green will be 2 per 5, which is 40%. And there's no leftover for the dead explant. So there is a presence of contaminated plate. While for the 15 minute of exposure time, there are none of them we have contaminated explant, so there's 0%, and 4 per 5 of the explant we are having viable and that is remain green, which is 80%, and remaining 1 per 5, which is 20%, is the dead explant. So there's no presence of the contaminated plate.
Okay, thank you, ANGELYN. So moving on to the first question. Okay, so based on the observation, explain the result obtained. Suggest a modification, if any, to improve the protocol. So who would like to try first?
I would like to try to answer the first question. So,
Okay, sure.
as we can see from, as we have been, based on the results, we can see that the number of the contaminated plants were exposed for 5 minutes is more than the plants that is exposed for 50 minutes. And moreover, only 40% of the explant that remains green that was exposed for 5 minutes, while 80% remains green for the 50 minutes. So, ways to improve the experiment or the protocol is, in my opinion, is by doing the experiment under a vacuum condition to avoid any air bubbles that will be formed for a more efficient sterilization.
I have another opinion for the modification. So since based on the observation, we can see that the contaminated explant is exposed to the 10% chloride solution for 5 minutes. It is found out to be higher than those who exposed for 15 minutes. And then the percentage of dead explant that's exposed to 10% chloride solution at 15 minutes is also higher than those at 5 minutes. So the hypothesis we can get here is the longer the exposure time of the explant to the sterilizing agent, the lower the percentage of contaminated explant found. So, but there's a problem if we get a prolonged exposure of the leaf to the sterilizing agent, that will kill the plant, because 20% of the explant from the leaf that is exposed to 10% chloride solution for 15 minutes is found out to be the dead explant based on the experiment just now. So the modification I suggest, maybe we can wash the explant with water and soap first. Like just now what Arief said, is the same purpose. We need to remove the air bubbles and like allow the water to interact with the plant tissue and give some exposure time to the sterilizing agent in the water.
Okay, thank you. Does everyone understand? Is there any question regarding? Okay. So everyone understands, or any question?
I think a good explanation from both of you. Thank you.
Okay, so moving on to our second question. Based on the result, draw a suitable conclusion. So anyone would like to try for this?
May I try again for the second question?
Okay, sure.
Okay. Based on the observation and the results that we have obtained, we can see that explants can be sterilized completely by immersing them in a 70% ethanol for 30 seconds and 10% Clorox for 15 minutes. So that means that the longer the exposure to the sterilizing chemical, on the other hand, will result in the explants that will be dead.
Okay. Now moving on to question number 3. Apart from exposure time, explain other factors that may affect the efficacy of the sterilizing agent used in this practical. Anyone?
Can I answer this question? Hello?
Go ahead.
Okay. Sorry. I think from this question, which is another factors or that affect the efficiency apart from exposure time, I think that, there are several factors that may affect the efficiency of the sterilizing agent, which are concentration of the sterilizing agent, the type of leaf surface, and the presence of organic material. So, for firstly, concentration of the sterilizing agent itself plays a role. Um, for example, the higher the concentration, the lesser time for a sterilizing agent takes to sterilize the plant. Then move to the next factor, which is type of leaf. For example, if we use a type of leaf that has more or of their surface is more lot of wax, for example, the leaf will absorb less amount of sterilizing agent at the leaf surface. So this might reduce the efficiency of the sterilizing agent used. So another factor that uh, is quite good to know, which is it is might due to the presence of organic material on the leaf surface, as the leaf were not rinsing properly during the experiment. So this can disrupt the efficiency, as the organic materials might come in contact and producing byproducts that reducing the efficiency of the sterilizing agents. So yeah, what do you guys think?
I think it's quite good, very good. Anyone has anything to add on? If no, then we move on to the next question. Question number 4. If surfactant is not used, predict the outcome of the practical based on the percentage obtained above. So anyone would like to try?
So can I try? Do you hear me?
Sure. Can.
Okay. So if the surfactant is not used, so surfactant is its function is to lowering the surface tension of the explant. So in the experiment we use twin 20. So if the surfactant is not used, so the percentage of the contaminated explant will become higher in both group in the 5 minutes and the 15 minutes experiment. So this is because without the surfactant, the sterilizing agent could not penetrate easily into the explant and expand due to the high surface tension of the leaf. So that's all.
Okay. Moving. Any question? No, then we move on to question number 5. If the step that treated the explant with 70% of alcohol was ignored, could the outcome of this practical be changed significantly? Explain. Okay, so I'll try for this question. I think yes, This is because the alcohol has antimicrobial properties by denaturation and dehydration of protein, and thus could remove the organic materials found on the surface of the leaves effectively. The immersion of the leaves in 70% of ethanol prior to immersion into sterilizing agent also further increases the efficacy of the sterilizing agent by removing air bubbles on the surface of the leaves. Do you guys know why? Okay, the reason alcohol could remove air bubble from the surface of the leaf is because it has lower surface tension than water, and it dehydrates the water so that the bubble can escape. Okay, any questions? If no, then I move on to the next question. Number 6, suggest a suitable sterilizing agent to replace Clorox used in this practical. Modify the protocol to suit the new sterilizing agent. I'll try for this also. A suitable sterilizing agent to replace Clorox used in this practical, I believe, would be hydrogen peroxide solution. We can use 12% concentration of hydrogen peroxide solution, and the exposure time of the leaf to the sterilizing agent is 5 to 15 minutes. And the precaution that we have to take is do not expose the hydrogen peroxide solution to the air for a long time, as it is relatively unstable and may decomposes to water and oxygen. What do you guys think? Hello. Do you guys have any other suggestion? If no, then at least give a response.
I have no other suggestion, but the suggestion from you is great.
Okay. Moving on to question number 7. Name the type of sterilizing agent used in this practical. Anyone?
So I will try this. So we use 10% Clorox in this experiment. So 10% Clorox is a halogen type.
Okay. Okay. Why is it halogen?
Can I try?
Okay, sure.
Is it because halogens have a greater absorption rate than any other sterilizing agents?
No. My question that I don't understand is why is the type of sterilizing agent used in this practical is halogen?
Ah,
I don't know if it's correct, but is it because 10% Clorox have a chlorine substance?
Exactly. Yes. Oh, understand now. Okay. Moving on to question number 8. Name the active ingredient of Clorox and its concentration at 10% solution of Clorox. Anyone?
Okay, I'll go for this. The active ingredient of the Clorox is the sodium hypochlorite at different concentration, which is a 0.53% at 10% of Clorox solution.
Okay. Any question? Do you guys understand how do we get the 0.53%?
No, I don't understand it.
Anyone would like to try to show why is it 0.53%? Okay. I try now. Why is it 0.53%? Because in Clorox solution, 100%, the commercial that we buy from the store, Clorox, 100%, it's actually, it contains 5.25% of sodium hypochlorite. And then in this practical, we only use 10% of that 100% Clorox. So of course, 1 cup of Clorox, and then you mix it in 9 cup of water. So the solution contain 0.53% or 0.525% of sodium hypochlorite. So that's how we get 0.53%. Okay,
Okay.
moving on to question number 9. Explain the procedure to prepare 10% Clorox solution. Mention whether you need to autoclave the solution or not and justify it. Anyone?
I think I can answer this. Okay. For this question, it's pretty straightforward because, of course, if we need to produce 100 ml of 10% of chloride solution, we need to dilute the amount of 10 ml of chloride solution in 90 ml of distilled water. So the solution in the question asks whether you need to autoclave or not. The answer is no. The solution does not need to autoclave, as the heat from autoclaving the solution will degrading the hypochlorous acid in the solution. So hypochlorous acid or HOCl is the chlorine residue from the reaction between sodium hypochlorite solution and water. So HOCl is a strong disinfectant that can penetrate cellular structure of microorganism easily.
Okay, thank you, Ame, for the good explanation. Do you guys have any question or anything to add on? Okay, moving on to question number 10. Justify the need to rinse and then expose the explant with sterile water for a certain period of time after Clorox treatment. So anyone would like to try?
I think I can try to answer the question.
Okay.
So based on the experiment, we can see that all of these experiments needed to be sterile at all conditions. So in my opinion, I think the need to use of sterile water is to wash away the sterilizing agent and to keep the explant from being contaminated.
Oh, okay. So basically is to prevent the contamination, the reintroduction of bacteria. Lah. Okay. Any other things to add on? If no, then we move on to the next question. If rinsing with sterile water was performed quickly without leaving the explant in water for a while, what would be the consequences? Predict based on percentage obtained above. So I would like to try for this question. The percentage of contaminated and dead explant will increase. This is because the sterilizing agent is not fully washed off from the explant. And then the sterilizing agent is very toxic to the plant, which results in dead explants because it dehydrates the plant. And then rinsing the explant with sterile water long enough is also important to prevent the reintroduction of microorganisms to the explant, just like what Arief just now said. Any questions? No? Then moving on to question number 12. If the source of the explant was not heavily contaminated, which part of the sterilizing protocol could be modified to make it more effective? And explain. Anyone would like to try?
So
Okay.
I will try. I'll try. Okay. So if the explant was not heavily contaminated, we can directly immerse the leaf in the 10% Clorox after the soaking the leaf with the twin 20 without sterilizing with the 70% ethanol. Because so ethyl alcohol, so alcohol is used for surface sterilization of explant as only the surface to dissolve the organic components. So if it was not heavily contaminated, so the amount of organic material may lower. So the 10% Clorox maybe can penetrate the leaf more easily. So it so that's all, I think. Or maybe we can lower the exposure time where we soak the 70% ethanol. Maybe in the experiment we use 30 seconds. Maybe we can lower it more. So that was my opinion. What do you think?
I think that is great. Anyone has anything to add on? Okay, no. Then any question? No. Okay. So move on to question number 13. If the source of the explant was sterile, which part of the protocol could be modified? Explain.
For my opinion, if the source of explant is originally sterile already, then I think the explant can be prepared directly. Like we can just directly jump to the cutting the explant into small pieces without the previous step like the sterilization and the cleaning step and the rinsing step, because it is originally sterile already. Then we just maintain the sterile condition of the plant, and then if we use other sterilizing agent and the disinfectant, then it will damage the structure of the plant also. So I think we can just proceed until the cutting process.
Okay. Thank you, ANGELYN, for the clear explanation. Anyone has any other question? If no, then we move on to question number 14. Suggest a method to grow a plant under sterile conditions for explant preparation. Anyone?
Okay, for this last question, I think there's a few methods to grow plant under sterile condition. But since it suggests a method, I think micropropagation can be done to grow a plant under sterile conditions. This is because the totipotency of plants can regenerate some shoots and roots. And during the culture, explant can be control their temperature and light in a culture room where the environmental factors cannot be disrupted by another environment. So for me, the best method is the micropropagation. I don't know about you guys, but I think that's the best method.
Okay. Anyone has anything to add on or any other question? No.
I have another opinion. Maybe my opinion is like quite simple. Maybe before we start the experiment, we need to make sure all the glassware is readily sterilized, and then we prepare the medium and we autoclave all the medium, and then we sterilize the surface of the explants and then transfer to the sterile jar so that it will grow into a plant later. Yeah, that's my opinion.
I see.
I have something to ask regarding the sterilize the surface of the plant. That means how sterilize? The one that we do in practical or how?
Yeah, it's still the same using the sterilizing agent, but just like I add in the sterile jar.
Oh, okay. So basically, okay, understand. Anyone has any other thing to add on? If no, then we'll end the discussion here. Thank you, everyone, and have a nice day.
Thank you.
Thank you,
Thank
everyone.
you.
Thank you, everyone.